Assalamu alaikum. We will continue our talk about proteins in Activity 5, Fate of Synthesized Proteins. At the end of this video, you will be able to notice that certain synthesized proteins are used in the cell and others are exported into the extracellular medium. To describe the journey of a protein being synthesized, and finally to describe exocytosis. From the beginning of this chapter, we've been talking about proteins, whether the sequence and structure starting from the simplest primary structure way to the complex quaternary structure. And we've said how much proteins are important in their function. For example, hemoglobin responsible for oxygen transport and insulin responsible for regulation of blood glucose. And how are these proteins synthesized? These proteins are synthesized by a process known as gene expression, which is divided into two phases, transcription and translation. During transcription, DNA is used as a template to produce messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in mRNA is used to produce a protein. However, is this the end of the story? Is this polypeptide produced now active after translation? Or does it need more modification by other cell organelles? Let's concentrate in this video in order to find the correct answer. In order to solve our problem, let's perform a simple experiment, where the aim of this experiment will be to find the path a protein passes through in a cell. In this experiment, insulin-producing cells, also known as cells of Langerhans, are cultured in the presence of radioactive amino acid. What's the importance of having radioactive amino acid? Well, culturing those cells in the presence of radioactive amino acid will give us, after transcription and translation, a protein, an insulin protein which is radioactive. In this way, we can follow the radioactivity of insulin in the cell's organelles. Now let's see what are the results of this experiment. Hold your notebook and follow up. After 5 minutes of culturing the cells in a radioactive medium, the autoradiograph showed that the radioactivity of insulin appeared first in the rough endoplasmic reticulum. Then after 10 minutes, this radioactivity appeared in the Golgi complex. After 1 hour, this radioactivity appeared in something known as secretory vesicles. And finally, as you can see on this autoradiograph, radioactive insulin is found somewhere on the plasma membrane. Following up insulin in this experiment, we can see that the order of insulin presence in the cell is rough endoplasmic reticulum, Golgi complex, secretory vesicles, and the plasma membrane. So for now, we know that a protein in this experiment, insulin, is produced in the endoplasmic reticulum and then transported to the bloodstream. However, what is happening in between is still not clear. In order to find what's happening in the way between the endoplasmic reticulum and the extracellular medium, take your time to compare proinsulin, which is produced in the endoplasmic reticulum, and insulin, which is exported to the extracellular medium. As you can see, the main difference is that this C chain, the green one, is found in proinsulin, however, it's absent in insulin. So how does proinsulin become insulin? What is the function of the organelles involved in modifying insulin, and how is active insulin transported to the bloodstream? The answer lies in the two organelles in which the protein passed through in the first experiment, the Golgi complex and the secretory vesicle. So what is the function of the Golgi complex and this secretory vesicle? Let's start with the function of the Golgi complex. But first, I need you to watch this video and remember what you've taken in our first lesson concerning the function of the organelles of the animal cell. Did you remember now? The Golgi apparatus is the site of processing proteins in order to become active. And the first form of modification is the change of structure of the protein from the simplest primary structure into the 3D active structure. And since here in the Golgi a protein is being activated, then also in the Golgi proinsulin is becoming insulin. However, what is the type of modification here? Is it also structural? Looking carefully at the difference between proinsulin and insulin, we can say that the difference is not structural. The difference here is in the sequence of produced insulin, where 32 amino acids in this green chain are being removed from proinsulin in order to give the active insulin. So here we can say that the Golgi complex is responsible for activating the protein, either by changing its structure or changing its sequence. What about the function of secretory vesicles? Well, secretory vesicles have a role in trafficking proteins outside the cell to reach their destination by a process known as exocytosis. Watch the following video and try to understand the meaning of the two terms, trafficking and exocytosis. Concerning the first role of the vesicles in trafficking proteins, you can see that secretory vesicles are holding proteins from the Golgi complex throughout the cell. So trafficking means movement of proteins in the cell with the help of vesicles. Now, what is exocytosis? By definition, exocytosis is a process by which the contents of a vesicle, in this case proteins, are being released from the intracellular medium to the extracellular medium through fusion of the vesicle membrane with the plasma membrane. As you can see here, the membrane of the vesicle fuses with the plasma membrane. The two membranes become one in order to release the proteins to the extracellular medium, in this case releasing insulin to the bloodstream. And that's simply it. The vesicles traffic proteins to the extracellular medium by exocytosis. Let's return to the title of our lesson, Fate of Synthesized Protein. When studying the example of insulin, we found that insulin is produced in the endoplasmic reticulum, transported to the Golgi in order to be modified, and then transported to the extracellular medium by a process known as exocytosis. And this is the story of a protein inside the cell. However, here we have another question to ask. Are all proteins subjected to exocytosis? Remember when we said that hemoglobin is a protein responsible for oxygen transport in our bloodstream? And you know from previous years that this hemoglobin is found inside red blood cells. So this protein is not subjected to exocytosis, it remains inside the red blood cell. So the fate of this protein is to stay inside the cell. We can give other examples such as the proteins that are embedded inside the plasma membrane or the membrane of the mitochondria of the cell. So here to summarize what we've said, all synthesized protein in the rough endoplasmic reticulum are transported to the Golgi apparatus in order to be modified and activated, as shown right here. However, those synthesized protein can have two different fates. Some proteins are subjected to exocytosis or to be released to the extracellular medium by fusion of the vesicle with the plasma membrane. This image here represents the fusion of the vesicle with the plasma membrane. And some proteins remain in the cell, such as the proteins incorporated to the membrane of organelles, the mitochondria, for example, or as we said, hemoglobin in the red blood cells. As an assignment, please make sure to study the summary sheet and to solve exercise for page 72. This video is prepared and recorded by teacher Zahra Shari and supervised by teacher Ihsan Hassan. Thank you for your time.